Hi guys, welcome back to my channel. This is Dr. Rajeshwar from YR PharmaTube. In the previous video, we have learnt the oxidation reactions not catalyzed by cytochrome P450. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of drug metabolism and medicinal chemistry, click on the links given in the description box below this video. In this lecture, we shall try to understand the reduction reactions of phase 1 drug metabolism. At the end of this video, you will be able to understand the process of reduction and the different types of phase 1 reduction reactions, which include reduction of carbonyl compounds, reduction of nitro and azo compounds, azido reductions, reduction of sulfur containing compounds, reductive dehalogenation, and certain miscellaneous reduction reactions. Before going to understand the reduction reactions of drug metabolism, let's take some keynotes to reduction and its definition in chemistry. Substances that have the ability to reduce other substances are said to be reductive or reducing agents. These agents transfer electrons to another substance and is thus itself oxidized. Because it donates electrons, the reducing agent is also called an electron donor. Electron donors can also form charge transfer complexes with electron acceptors. See the animated figure in which sodium gives or donates one outer electron to fluorine, bonding them to form sodium fluoride. Thus, the sodium atom is oxidized and the fluorine is reduced. Thus, reduction is the transfer of electrons between species in a chemical reaction. It is a process of gaining electrons or a decrease in the oxidation state by an element. A reductive chemical reaction involves increasing electrons associated with a single atom or a group of atoms. In drug metabolism, reduction reactions are the second type of phase 1 reactions. Reduction reactions are less commonly encountered than oxidation reactions. Yet, these reduction reactions are very important group of biotransformations where certain compounds whose functional groups that are reduced are naturally in a higher oxidation state. These functional groups such as carbonyl, nitro, azo groups, etc. tend to be biotransformed by reduction to other functional groups such as amino and hydroxyl or alcoholic groups. These products of the reduction reactions subsequently undergo further phase 2 conjugation reactions to form derivatives that are more water soluble than the original drug. That is why it is likely that cytochrome P family of enzymes carried out reductions well before they evolved to oxidize. The enzymes used to catalyze these metabolic reduction reactions are usually specific in their action. For example, carbonyl groups such as aldehydes and ketones are reduced by soluble aldoketo-reductases, nitro groups by soluble nitro-reductases, and azo groups by multiple component microsomal reductases. All these enzymes are found mainly in the liver but also occur in the kidneys and other tissues. Many of these enzymes require NADPH as a coenzyme. The 6 different types of reduction reactions include number 1, reduction of carbonyl compounds, number 2, reduction of nitro and azo compounds, number 3, azido reductions, number 4, reduction of sulfur containing compounds, number 5, reductive dehalogenation, and number 6, miscellaneous reduction reactions. Number 1, reduction of carbonyl compounds. Many drugs contain carbonyl group in their structures and undergo reduction reactions, especially the ketonic group. Aldehydes and ketones are widely distributed and have several biological functions. In addition to alcohol dehydrogenase, there are several enzymes in the aldoketo reductase family that may participate in the metabolism of aldehydes and ketones in the kidney. The carbonyl group is reduced through a reduction reaction that is catalyzed by aldoketo reductase enzymes. These enzymes present in the liver and other tissues like kidney which require NADH or NADPH as a cofactor. Oxidoreductase enzymes can also reduce aldehydes and ketones. As carbonyl groups are of aldehydic and ketonic, let us discuss the reduction reactions individually according to the functional group present in the drug molecule. Reduction of aldehydes. Aldehydes undergo reduction to form primary alcohols. Phase 1 metabolites formed by oxidative deamination of drugs also contain carbonyl group. Aldehyde metabolites are easily oxidized to carboxylic acid and occasionally reduced to primary alcohols. Aldehydes can also be converted to alcohols but can undergo additional pathway of oxidation to carboxylic acids. The alcohols produced by the reduction reactions can be eliminated by glucuronidation or other phase 2 processes. For example, chloral hydrate undergoes reduction to form its corresponding alcohol derivative, that is trichloroethanol. Reduction of ketones. Ketones generally resist metabolic oxidation reactions, but a large number of aromatic and aliphatic ketones are reduced to the corresponding secondary alcohols. In the case of ketones, although the reduction is reversible, ketoreductase utilizes NADPH, the concentration of which is higher than NADP+, and this drives the reaction toward the secondary alcohol. Similar to the aldehydes, ketones also undergo further conjugation reactions, for example, glucuronidation. These reduction reactions are frequently stereospecific, which means that reduction of ketones lead to the creation of an asymmetric center, that is the chiral center, resulting in two possible stereoisomeric alcohols. For example, reduction of acetophenone leads to enantiomeric alcohols S and R isomers of methylphenylcarbinol with the S isomer predominating. The mechanism involves a hydride transfer from the reduced nicotinamide moiety of the cofactor NADPH or NADH to the carbonyl carbon atom of ketone. Often, ketone reduction yields alcohol metabolites that are pharmacologically active. Achiral drugs containing ketone groups such as oral hypoglycemic acetohexamide usually give predominantly one enantiomer on reduction. Acetohexamide is metabolized rapidly to give active metabolite S-hydroxyhexamide. When chiral ketones are reduced, they yield two possible diastereomeric or epimeric alcohols. For example, the R plus warfarin, an anti-clotting agent, undergoes reduction of its side chain ketone group to generate the RS plus alcohol as the major metabolite. Small amounts of the RR plus diastereomer are also formed. In contrast, the S-warfarin undergoes little ketone reduction and is primarily hydroxylated in aromatic ring. Reduction of alpha-beta unsaturated ketones result in reduction not only of the ketone group but of carbon-carbon double bond as well, which means that alpha-beta unsaturated ketones are typically metabolized to saturated alcohols. For example, norethindrone, a synthetic progestin found in many oral contraceptive drug combinations, undergoes metabolic reduction to give 3-beta, 5-beta tetrahydrometabolite. Ketones resulting from metabolic oxidative deamination processes are also susceptible to reduction. For example, amphetamine is deaminated to phenylacetone, a non-aromatic ketone which is subsequently reduced to 1-phenyl 2-propanol. Number 2. Reduction of nitro and azo compounds. The reduction of aromatic nitro and azo xenobiotics leads to aromatic primary amine metabolites. The reduced compounds or metabolites are conjugated and eliminated from the human body. Reduction of nitro compounds. Nitro groups can be reduced all the way to amines. Aromatic nitro compounds are reduced initially to the nitroso and then to hydroxylamine intermediates as shown in the metabolic sequence. This reaction is catalyzed by NADPH dependent hepatic nitroreductases present in the liver. The first step requires an enzyme such as cytochrome P450 or anaerobic bacteria, but reduction of nitroso groups is so facile it is usually a simple chemical reduction mediated by biological reducing agents such as ascorbate or NADPH. Although the pathways are shown as 2-electron oxidations and reductions, 1-electron chemistry can also occur. There are not many drugs that contain a nitro group and most of them are toxic compounds. This reaction can have toxicological significance as nitro groups can be reduced to nitrosoarenes and hydroxylamines, and this is thought to be reduced to major pathway towards the carcinogenic effects of aromatic amines. Here is an example of 7-nitrobenzodiazepine derivatives clonazepam and nitrazepam, which undergoes metabolic enzymatic reduction to their respective 7 amino metabolites in human. The skeletal muscle relaxant dantrolene also reportedly undergoes reduction to aminodantrolene in humans. Reduction of azo compounds. The 4-electron reduction of the azo group leads to the cleavage of the molecule and the production of 2 amines. Azo compounds are susceptible to reduction initially produces hydrozo intermediates which are then reductively cleaved into the appropriate amines. Thus, azo reduction is believed to proceed via a hydrozo intermediate. This hydrozo intermediate is subsequently cleaved reductively to yield the corresponding aromatic amines. In addition, bacterial reductases present in the intestine can reduce nitro and azo compounds, especially those that are absorbed poorly or excreted mainly in the bile. Reduction of azo group is catalyzed by cytochrome P450 enzyme and by NADPH cytochrome P450 reductase under anaerobic conditions. Bacterial azo reductase in gastrointestinal tract mediates this type of reaction. The enzymatic reduction of azo compounds is best demonstrated by the conversion of prontosil to the active sulfanilamide metabolite in the liver. This reaction has historical significance for it led to the discovery of sulfanilamide as an antibacterial agent and eventually to the development of many of the therapeutic sulfonamide drugs. Bacterial reductases present in the intestine play a significant role in reducing azo drugs, particularly those that are absorbed poorly. Accordingly, the two azodies, tetrazine and amaranth, have poor oral absorption because of the many polar and ionized sulfonic acid groups present in their structures. Therefore, these two azo compounds are metabolized primarily by bacterial reductases present in the intestine. Number 3. Azido reduction. Azides may be converted to amines by cytochrome P450 isoenzymes and NADPH cytochrome P450 reductase. The anti-HIV drug zidovudine undergoes reductive reaction to a toxic metabolites 3-amino 3-deoxythymidine. Number 4. Reduction of sulfur-containing compounds. Reduction of sulfur-containing functional groups such as that sulfoxide and disulfide moieties also constitute a major reductive pathway. Reduction of sulfoxides. Although sulfoxide functionalities are oxidized mainly to sulfones, they sometimes undergo reduction to sulfides. The importance of this reductive pathway is seen in the metabolism of the anti-inflammatory agent sulindac. Sulindac undergoes reduction to an active sulfide metabolite that is responsible for the overall anti-inflammatory effect of the parent drug. Sulindac or its sulfone metabolite exhibits little anti-inflammatory activity. Reduction of disulfides and other oxidation states of sulfur. Disulfides can be reduced to 2-thiols. The best example is the reduction of oxidized glutathione, which is a disulfide, is reduced to 2 molecules of thiols, and the reaction is mediated by glutathione reductase enzyme. In addition, exchange can occur with other thiols mediated by protein disulfide isomerase. In the other example, reductive cleavage of the disulfide bond in disulfiram yields N,N-diethyl dithiocarbamic acid as a major metabolite. In the third example, sulfide formation involves the reduction of dimethyl sulfoxide, abbreviated DMSO, to dimethyl sulfide. DMSO is metabolized to a minor extent by this reductive pathway. The characteristic unpleasant odor of dimethyl sulfide is evident on the breath of patients who use this agent. Sulfonic acids can probably also be reduced back to the thiols, but because of the reactivity of the sulfonic acid, this is not generally observed. Some other oxidation states of sulfur can be reduced, but not sulfonic acids or, as mentioned above, the sulfones. Number 5. Reductive dehalogenation. A 1-electron reduction of the bond between an aliphatic carbon and a halogen leads to a halogen anion and a carbon-free radical. A good example is the reduction of carbon tetrachloride. The first product in the reduction is the trichloromethyl free radical. Carbon-centered radicals are not very reactive with biological molecules, but they react very rapidly with molecular oxygen, that is a diradical, to form a peroxy-free radical, which is a quite toxic compound. The ease of reductive dehalogenation depends on the degree of electron deficiency of the drug molecule. For example, halothane is somewhat more difficult to reduce than carbon tetrachloride, and in general, reductive dehalogenation does not occur with a monochloro compound. Under aerobic conditions, halothane, an anesthetic agent, undergoes reductive dehalogenation to form 2-chloro-1,1-difluoroethene by cytochrome P450 enzyme. The intermediate product 1-chloro-2,2,2-trifluoroethyl radical, however, is either further reduced to 2-chloro-1,1,1-trifluoroethene or covalently bind with hepatocytes, resulting in hepatitis. It is to note that halothane may also undergo oxidative dehalogenation by a different mechanism. Most reductive dehalogenation reactions are likely mediated by cytochrome P450, in which the halogenated compounds compete with molecular oxygen for its reduction. Therefore, such reduction occurs more readily under hypoxic conditions, that is, when the body tissues are in low levels of oxygen. And number 6, miscellaneous reduction reactions. Several minor metabolic reductive reactions also occur in xenobiotics. Reduction of N-oxides to the corresponding tertiary amine occurs to some extent. This reductive reaction is significant because several tertiary amines undergo oxidation to form polar and water-soluble N-oxide metabolites. If reduction of N-oxide metabolites occurs to a significant extent, drug elimination of the parent tertiary amine is hindered. N-oxide reduction is assessed by administering the pure synthetic N-oxide in vitro or in vivo and then detecting the formation of the tertiary amine. For example, imipramine N-oxide undergoes reduction. Amine oxides are readily reduced back to tertiary amines. There are a few drugs that are amine oxides, but there are many drugs that are tertiary amines, and amine oxides are common metabolites. The amine oxide is often pharmacologically inactive, however, because they are readily reduced back to tertiary amines, amine oxides can act as a buffer to the concentration of the tertiary amine. This is the list of references followed for the lecture. That's all in this video, the phase 1 reduction reactions of drug metabolism. In the next lesson, we will discuss about the hydrolysis reactions of drug metabolism. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.